Assalamualaikum. Let's continue with the part 5 of the chapter 10 lecture. So, mechanisms that prevent self-fertilization. So, in many angiosperm, they have mechanisms that make it difficult or even impossible for a flower to fertilize itself. For example, as shown in this figure, Oxalis alpina, or known as alpine wood sorrel. This plant produces two types of flower on different individuals known as trump flower with short style and long stamen, and also pin flower with long style and short stamen. So when insect hunt for nectar, pollens get collected on its body, and then trump pollen will be deposited on pink stigmas and vice versa. So they cannot fertilize their own self. This is another example. Sagittaria latifolia, commonly known as arrowhead. These are dioecious plants that produce only staminate flowers or carpellate flowers on the right. So they have different features. So in preventing self-fertilization, the most common anti-selfie mechanism in flowering plants known as self-incompatibility, or the ability of a plant to reject its own pollen. So researchers are unraveling the molecular mechanisms that are involved in self-incompatibility. So this is the illustration of a self-incompatibility and interspecific incompatibility, or also known as interspecies incompatibility in Brassica species. So Si denotes as self-incompatibility, C as compatibility, and ISI as interspecific or interspecies incompatibility. So for self-incompatibility, so the pollen from this anther cannot fertilize the same flower. However, within the same species, for example, Brassica rapa, pollen from the anther of one flower can fertilize and can stick to the stigma and develop pollen tube and fertilize the other flower from the same species. However, if it's from interspecies, for example, Brassica rapa and Brassica oleracea, it is known as interspecies or interspecific incompatibility. Even the pollen from the anther of another flower cannot fertilize the other flower. Even though from two different flowers, but it is interspecies, it cannot fertilize. So for self-incompatibility, so some plants reject pollen that has S gene, matching an allele in the stigma cells. So recognition of self-pollen trigger a signal transduction pathway leading to a block in growth of pollen tube. So we can see here the S gene is known as S1 and S2 for the flower. So what happens if the pollen comes from the flower with the same S gene, S1 and S2? It blocks the formation of the pollen tube. Okay, incompatible. However, as shown in the middle figure, so as pollen with S1 cannot form pollen tube, but pollen of with S3 gene can form pollen tube because it is not similar to S1 and S2. It only rejects if the S gene is similar, matching the allele in the stigma cells. as shown as this third figure. S1 and S2 allele in the sigma cell is compatible with the S3 and also S4 gene of the pollen. So both can produce pollen tube and fertilize the egg. So for other terms, dichogamy is known as stamen and carpels mature at different times, and dioecious plant is where male and female flowers on different plants. So this is another illustration of self-incompatibility. S1 and S2 allele with matching S gene on the pollen cannot produce pollen tube. However, if different allele S3 and S4, it can produce pollen tube and fertilize. Fertilization can happen with the S1 and S2 allele in the ovary. For double fertilization, so after landing on a receptive stigma, a pollen grain germinates and produces a pollen tube that extends down between the cells of the style towards the ovary. So the pollen tube discharges 2 sperm into the embryo sac. So in double fertilization, only 1 sperm fertilizes the egg. What happened to the another sperm? So the other sperm combine with the polar nuclei, giving rise to the food storing endosperm. This is a with triploid nucleus. Growth of the pollen tube and double fertilization. So number 1, if a pollen grain germinates, a pollen tube grows down the style towards the ovary. So the pollen grain land on the stigma and then it will grow the pollen tube, bringing 2 sperm towards the ovary. So the pollen tube discharges 2 sperm into the female gametophyte or the embryo sac within an ovule. So this is the ovule. It has the female gametophyte. So the sperm will be discharged here near the egg. So one sperm will fertilize the egg, forming a zygote, and the other sperm will combine with the 2 polar nuclei in the middle of the embryo sac. So this is 2 polar nuclei of the embryo sac, large central cell. This will form triploid cell that will then develop into nutritive tissue. So this tissue is called endosperm. Development from ovule to seed. So a seed consists of an embryo, which is the fertilized egg, a nutrient supply, this is the nutritive tissue, which is the endosperm, and a seed coat, this is the protective layer. So this seed coat is the outermost layer, and then here got the endosperm layer, and in the middle is the embryo with the shoot apex and root apex with 2 cotyledons, or it can be also 1 cotyledon. So after double fertilization, each ovule develops into a seed. So the ovary develops into a fruit enclosing the seed. So usually when we cut open a fruit, we will find the seed in the middle. For embryo development, the first mitotic division of zygote is transverse, so splitting the fertilized egg into a basal cell and terminal cell. So as we can see here in the ovule, here lies the zygote. So the zygote will become terminal cell and also basal cell. This will further develop into proembryo, suspensor and basal cell, and then this will eventually becoming the seed, the whole seed with the seed coat, the endosperm, with the suspensor, root apex, shoot apex, as well as the cotyledons. So the endosperm development usually precedes the embryo development. In most monocots and some eudicots, the endosperm stores nutrient. So this nutrient will then be used by the seedling after germination. So in other eudicots, the food reserve of endosperm are completely exported to the cotyledons. So the embryo and its food supply are enclosed by a hard protective seed coat. So here we can see the structure of a mature seed. So in common garden bean or a eudicoid, a dicoid, embryo with two cotyledons. So each side got one cotyledon. So the embryo consists of the hypocotyle, radical, and thick cotyledons. So this figure shows a common garden bean, a eudicoid with thick cotyledons. The fleshy cotyledons store food absorbed from the endosperm before seed germinates. The seed of other eudicoids, for example, castor bean. So castor bean has thin cotyledons. The narrow membranous cotyledon shown in the flat edge flat views, okay, absorb food from the endosperm when the seed germinates. So this is the cotyledons. This is the cotyledons of the castor beans. The embryo of a monocot has a single cotyledon, a coleoptile and coleorhiza. So maize, so this is the seed of a maize. A monocot. Maize has only one cotyledon, single cotyledon. And maize and other grasses have a large cotyledon called scutellum. So the rudimentary shoot is sheeted in a structure called the coleoptile. So here is the coleoptile, and this is the rudimentary shoot, and the coleorhiza covers the young root. So here is the structure of the coleorhiza. So we will continue the development from ovary to fruit in part 6 of the lecture. Thank you.